My name is Joel Ross, and I'm a research associate in the laboratory of Dr. James Ellis at the Hospital for Sick Children. And today I'm going to be talking about some of the work that we've done on identifying neuronal phenotypes associated with neurodevelopmental disorders such as Rett syndrome using iPS cell-derived neurons. So just a brief overview of what I'll be discussing here. First, I'll just introduce Rett syndrome and some of what we know about it and why we were interested in using this in order to begin our investigations of neurodevelopmental disorders. Second, a brief discussion of some of the experimental considerations when it comes to planning and implementing a study aimed at identifying neuronal phenotypes using iPS cells, and then a discussion of some of the phenotypes that our groups and other groups have used in order to learn more about Rett syndrome. So very briefly, this is a schematic representation of the approach that our lab and many other labs use, which was published in a review by Marchetto and Gage. And so we generate iPS cells from individuals with specific neurodevelopmental disorders and, when possible, from their unaffected relatives. And we induce these iPS cells to differentiate into neural precursors in the neurons. And our primary aim is to study these neurons in order to identify specific cellular phenotypes, differences in the way that they function in comparison to neurons from unaffected controls, in order to learn more about these disorders. And eventually, we hope these phenotypes can serve as the foundation for drug screens aimed at identifying potential novel therapeutics. So Rett syndrome is a severe neurodevelopmental disorder that is almost exclusively seen in females. The symptoms appear first at around 6 to 18 months of age, and they include these symptoms include a loss of acquired language, intellectual disability, and a number of different behavioral phenotypes, as well as breathing and eating difficulties. The vast majority of cases of Rett syndrome are caused by mutation of the gene MECP2, or methyl CpG binding protein 2, which is found on the X chromosome. MECP2 is a very well-studied nuclear protein, but exactly what it does in neurons is still not very well understood. What we do know is that MECP2 is able to bind both methyl CpG and hydroxymethyl CpG motifs present in DNA, and when recruited to chromatin, it's able to both activate and repress the expression of its target genes. And although we are still learning more and more about which genes MECP2 actually targets, what we do know is that global misregulation of neuronal gene expression that results from a loss of MECP2 profoundly alters neuronal function. And so in addition to studying Rett syndrome simply from the perspective of wanting to learn more about this disorder, Rett syndrome is viewed by many as a very interesting initial proof of principle for establishing the feasibility of iPS-derived neurons for modeling neurodevelopmental disorders. And the reasons for this include the fact that most cases of Rett syndrome are caused by mutation of MECP2, and because of the fact that the phenotypes are quite severe. But the main reason is because MECP2 has been knocked out in mice, and we know that these mice recapitulate Rett syndrome and many of the associated phenotypes. And as a result of studying these mice, we know what phenotypes we can expect to see in iPS cell-derived neurons from people with Rett syndrome. And these include decreases in both the size and complexity of neurons, a decrease in the number of excitatory synaptic connections, as well as impairments in other aspects of neuronal function, including action potential firing. And so therefore, by knowing what phenotypes to look for, we can really use this disorder in order to try to establish a baseline for the effectiveness of iPS-based models for these neurodevelopmental disorders. And so in the data that I'm going to be discussing later on in subsequent slides, we're using iPS cells from 2 individuals, or at least for the work done in the laboratory of Dr. Ellis. These individuals include one female who has a disruption of exons 3 and 4 of one of her MECP2 alleles, as well as a second individual who has an 11 base pair deletion within exon 1 of MECP2 that specifically disrupts the E1 isoform of MECP2, which is the dominant isoform within the brain. So before we start doing experiments in order to try to identify cellular phenotypes, there are a number of different considerations that we have to be mindful of when planning an experiment. So in order to identify cellular phenotypes, we need iPS cells from both individuals with the disorder of interest, but also unaffected individuals. And in addition to this, we need to be thinking in terms of how many lines do we need? How many individuals do we need? Much of the published literature is based upon comparing one unaffected individual to an unaffected individual, but more and more studies are trying to expand into including multiple individuals with disruption of the same gene, and particularly in including multiple unaffected individuals. And when we talk about the experimental controls that are used, ideally, we're working with iPS cell or pluripotent cell-derived neurons from unaffected individuals. Many studies have used human embryonic stem cells as the control. There are a number of benefits to doing this, particularly when working with the well-studied lines. But we find the idea of using iPS cells from unaffected individuals to be quite attractive because we know that these individuals can develop to adulthood without a manifestation of some of the disorders we're interested in, such as autism spectrum disorder, which is the focus of other projects in the lab. And ideally, because of the nature of studying people, which are a naturally outbred population, it can sometimes be beneficial to generate iPS cells from the unaffected relatives of some of these individuals. But an ideal control for experimental studies aimed at understanding the function of an individual gene, isogenic controls are very, very helpful. And these include, in particular cases where you're studying female iPS cell lines with an X-linked disorder in mind, which I'll talk about in the next slide, but also of great interest right now is the use of genome editing technologies in order to either introduce or correct specific mutations in genes of interest. So when I talk about isogenic controls, one potential source of isogenic control pluripotent cells is from females who carry an X-linked mutation. And the reason why we're able to do this is because iPS cells developed in our lab and many other labs exhibit line-specific clonal X chromosome inactivation status, which is all based upon the X chromosome inactivation status of the original reprogrammed cell. And so, for example, we begin with fibroblasts for many of our studies. If the primary fibroblast that was infected to give rise to a line had its active X encodes the wild-type MECP2 gene, the result is an iPS cell line that expresses MECP2 and acts as a wild-type isogenic control. If the original reprogrammed fibroblasts expressed only the mutant allele, then the result is an iPS cell line that acts as an isogenic mutant. And by comparison to these cells, we can really gain precise impact or insights into the function of the particular gene of interest. And although this slide does not translate well because of an editing issue, what I'm showing here is just a brief discussion of human genome editing technologies and how they can be applied to the study of disease using pluripotent cells. Genome editing is essentially using one of three major technologies, be they TALENs, CRISPRs, or zinc fingers, in order to introduce double-stranded breaks into specific genomic loci, and then using those double-stranded breaks to facilitate homologous recombination, which human cells are notoriously inefficient at accomplishing, or at least historically. And by using this approach, we can now introduce specific mutations into iPS cells or correct mutations. So therefore, genome editing allows us to generate isogenic pairs where mutations can be introduced into control cells or disease-associated mutations can be repaired in cells from individuals with specific disorders. So very briefly, this is a schematic representation of the differentiation protocol that that my own work and much work in the Ellis lab has employed. This is a modification of a protocol published by Fred Gage's group with Kristen Bernand as the first author. And briefly, we begin with the cells growing in feeder-free adherent conditions. We lift the cells and allow them to develop into embryoid bodies in the presence of dual SMAD inhibitors to force specification to the ectoderm lineage. We seed these cells on polyornithine and laminin and allow them to form neural rosettes, which are indicative of successful differentiation to the neuroectoderm lineage. We then manually dissect these neural rosettes and dissociate them with Accutase to generate neural precursors, which we can then grow for multiple passages in vitro in order to cryopreserve the cells, which greatly facilitates downstream experiments, or we can culture them under differentiation conditions to allow them to differentiate into neurons. For gene expression experiments, we will routinely work with relatively young neurons, which have been grown in culture for 3 to 4 weeks. But for analyses of synaptic connectivity, we tend to allow the neurons to mature in vitro for 2.5 to 3 months. And if synaptogenesis is the aim, we also include primary astrocytes. A number of groups have found that inclusion of astrocytes greatly improves the formation of synapses, and most groups are either using primary mouse or rat astrocyte cultures or primary human cultures, and our lab has experience using both, and both will facilitate the formation of synapses. And before moving on to discussions of the actual experiments, we should also address the potential for heterogeneity within iPS cell-derived neurons. At every step of the differentiation process, there is the potential for generating undesired cell products. And it's best to know which cell products are being made and which cell products are present within your final cultures, which will then greatly facilitate the interpretation of any experimental results. So examples of potential heterogeneity include neural crest-like stem cells, which can arise during the rosette formation stage and can sometimes contaminate the neural precursor populations. The neural precursors themselves are able to make several different lineages. They can make neurons, they can make astrocytes quite efficiently, and in some cases they can also make oligodendrocytes. Another potential problem with the neural precursors is their ability to continue to proliferate after, even after the growth factors have been withdrawn and trophic factors have been supplied. Particularly, they can sometimes undergo several rounds of continued proliferation, which can then dramatically alter the overall density of cells for the final experimental analyses, which can then also alter the final experimental analysis. And another consideration is the fact that NPCs generate neurons that are in and of themselves often a heterogeneous population. Multiple neurotransmitter types are often generated from the same differentiation. For example, the production of both glutamatergic and GABAergic neurons, and also the regional identity of neurons can vary to some degree, regardless of what the overall aim of the differentiation protocol is. And finally, another consideration is line-to-line variability, which is one of the reasons why it's very important to consider using multiple lines from each individual understudy, so multiple independent iPS cell lines. Once the neural precursors and neurons are generated, it can be very valuable and important to begin to characterize these cultures. So this, what I'm showing here is an example of an immunocytochemistry-based characterization of some neural precursors, because the disorders that we study, Rett syndrome and autism spectrum disorder, are disorders of cognitive function. We're very interested in examining the cerebral cortex and examining the function of excitatory cortical neurons. And so that is our aim in our differentiation protocol. And here are examples of successful stains with markers of excitatory cortical neural precursors, including FOXG1, OTX2, and FORCE1. Another widely used marker is PAX6, which I'm not showing here. Nestin and Musashi are often used as markers of neural precursors. And for a more precise marker of our desired neural precursor cell type, we often stain the cells with SOX1, which is an indicator of successful differentiation to central nervous system NPCs, ruling out, for example, neural crest-like cells. Another approach to characterizing the resultant populations of cells include looking at the RNA level. And so here I'm showing an example of experiments that were done by the Ellis Lab, published in a recent paper in Neurobiology of Disease, where we performed single-cell gene expression analyses using fluidigm technology. And particularly, we are able to sort individual live neurons and obtain RNA for looking at the expression of 48 genes in 48 cells per array. And when we compile all these data, here what you can see are the number of cells that were analyzed that expressed a specific marker. We can look at the expression of certain markers of both neural progenitors and neurons, including a number of different neurotransmitter phenotypes. And all of these primer sets and this approach is described in a study from Ricardo Dolmesh's lab that is referred to in the final slide. And so once we start compiling some of these data, we can, for example, look at the successful generation of specific cell types. So for example, by looking at a number of very specific markers here, again, described in this paper and in Ricardo Dolmesh's paper, we have found that we are successfully generating cells with a cortical forebrain identity, including a mix of cells from both the lower and upper layers of the cortex. And we're also generating a mix of cells that are both inhibitory or excitatory in nature, suggesting that we are able to recapitulate several aspects of cortical neuronal development in a dish. And so once we have our neurons and we have some idea of what kinds of neurons we're making, now we can start to consider looking for specific cellular phenotypes associated with the disorders of interest. And so again, I'm going to be focusing on analyses of gene expression, particularly those performed by Rudolf Janish's lab in a relatively recent paper, and functional assays of the neurons, as well as looking at the morphology of these cells. And particularly when it comes to Rett syndrome, we're very interested, once these phenotypes are identified, to determine if they can actually be rescued by either genome editing or, in the case of these experiments, reintroduction of the MECP2 gene. And upon determining that any of these phenotypes actually can be rescued, this points towards the possibility of these phenotypes potentially being used for drug screens in the future. And so first, what I'm showing here from the perspective of looking at gene expression phenotypes in neurons, I'm highlighting some really stunning work that was done by Rudolf Janish's lab and the gene expression analyses done in collaboration with Rick Young's group, also at the Whitehead Institute. And what they are focusing on in this study is human embryonic stem cells, the WIBR1 and WIBR3 ES lines, and which have been genome edited using TALENs in order to disrupt the MECP2 gene. So these are the mutant versus control cells. And very interesting observation that they made by looking at gene expression in these cells was that the overall levels of mRNA expression of MECP2 in ES cells rises quite dramatically in the differentiation to neural precursors, but then it levels out in the transition from neural precursors to neurons, which is quite interesting considering the fact that MECP2 protein, at least in the mouse system, is typically associated with postmitotic neurons. But when they use the same system in order to look at overall protein levels, what they found was that the protein levels are what is changing quite dramatically between neural precursors and neurons. And although some of this was known previously from studies in the mouse, it's these types of approaches that can be used in order to characterize the function of other genes of interest relating to other neurodevelopmental disorders, and points towards the power of this system for looking at expression of genes at specific developmental time points. But from a global perspective, this work provided some really interesting insights to the molecular basis of the neuronal dysfunction observed in Rett syndrome. And one critical aspect of this work was the observation that especially with 4-week-old neurons, the amount of RNA present in each individual neuron drops quite substantially between mutant and control cells. And this, along with the fact that mutant cells are known to be smaller, as I'll show you in a subsequent slide as well, made it so that precise analyses of global gene expression required very specific controls, which are elaborated upon in this study, but they include, for example, the inclusion of specific RNA spike-ins that allowed more precise analyses. But with these controls in mind, what the Janisch and Young labs found was a profound alteration of very basic gene expression pathways within these neurons. For example, misregulation of of transcriptional regulation of mRNA processing and of translation, suggesting that these profound neuronal phenotypes are actually the result of quite profound transcriptional and gene expression dysfunctions. And these types of approaches can also be used to study other neurodevelopmental disorders. From the perspective of functional assays, we and many others are very interested in synaptic connectivity. And so as a first step in determining whether our human iPS cell-derived neurons are forming synapses in a dish, we performed a number of immunocytochemistry experiments. And here's an example of one such experiment where in this cartoon on the left, what I'm showing is a schematic representation of the readout of the experiment. I have labeled these cells with antibodies against synapsin 1, which marks axon terminals, PSD95, which labels the postsynaptic portion of excitatory synapses, and where we see an overlap between red and green signals along a MAP2 positive dendrite, this is evidence of the formation of an excitatory synapse. But the ideal way to study synapses is to look at them from the level of function. So all of the electrophysiology work that I'm going to present here was done in collaboration with Dr. Michael Salter's lab at the Hospital for Sick Children, and the recordings were done by Wenbo Zhang, a very talented postdoc in his group. And so this is an example of a trace of synaptic activity, specifically excitatory synaptic activity within these cultures. The readout here is called miniature excitatory postsynaptic currents. And essentially what Wenbo has done is he is recording all of the excitatory electrical information that is entering the neuron that he's interested in. And all of this excitatory information is plotted here as these synaptic currents. And Wenbo can analyze not only the number of synaptic events, but also the size of the events, which can give us insights into any neuronal dysfunction that occurs. And so when we look at the size of the events or the amplitude of these individual synaptic events, Wenbo found that there was a modest but statistically significant and consistent decrease, suggesting decreases in the actual strength of synapses. But a more profound phenotype was the frequency of excitatory events, which is correlated with several factors, but one being the actual number of excitatory synapses that are being formed. And again, this recapitulates what is known or what is expected from studies in the mouse system within our human iPS cell-derived neurons. Other tools for studying the function of neurons include calcium imaging. Here what I'm showing is an experiment that I did where I performed live imaging, and over a period of 1 minute, which is shown here, I, at 30 seconds, treated the cells with 55 millimolar potassium chloride, which depolarizes the neurons and opens voltage-gated calcium channels, resulting in a swift influx of calcium, which is indicated particularly between 31 and 33 seconds, with a dramatic rise in the fluorescence intensity emitted by this fluorescent calcium indicator, fluo4AM. But other groups, particularly in this case, the groups of Fred Gage and Alison Moatri, have used calcium indicators in order to monitor action potential firing. In response to firing an action potential, there is a transient and very strong influx of calcium into individual neurons. And this is indicated here in a trace from 1 neuron. And here we're showing traces from 23 neurons. And each one of these spikes is thought to represent an action potential firing. And what you can see when comparing traces from wild-type control iPS cells to those of individuals with Rett syndrome is that there is a profound decrease in the number of action potentials being fired and in the ability of individual neurons to fire action potentials. An alternative approach to studying action potential is the use of multi-electrode arrays, for example, again, in this paper from Rudolf Janish's lab, where essentially the neurons are seated on a plate that has electrodes present which can record the spontaneous electrical activity of the neurons. And so here, for example, between days 48 and 55 in vitro, these neurons begin to start robustly firing action potentials as recorded by these arrays. And when these data were actually plotted, what was observed was that compared to control neurons, the MECP2 null neurons that were generated had a profound decrease in their ability to fire action potentials. Our group has also studied action potential firing. Again, this is electrophysiology done by Wenbo Zhang in Mike Salter's lab, where Wenbo has used electrophysiology to record action potential firing, particularly when he has evoked action potentials by injecting current into the neurons. And here is an overlay of many traces from wild-type neurons from an unaffected control compared to neurons from an individual with Rett syndrome. And most notably, what you can see is that as you increase the current injected into a neuron, control neurons will continue to fire more and more action potentials. But in neurons from individuals with Rett syndrome, this appears to taper off, and there's a plateau of the ability to fire evoked action potentials. And one of the benefits of using electrophysiology is that there are a number of different parameters that can be analyzed in these experiments. And Wenbo was actually able to correlate this decrease in action potential firing with a decrease in the currents of voltage-gated sodium channels, which play an extremely important role in action potential firing, so therefore providing perhaps a little bit more mechanistic insight into the phenotypes that are observed. Another electrophysiological phenotype that was observed in these studies was a decrease in the capacitance of individual neurons. And this is particularly notable because capacitance is correlated with cell size. And we know from work in the rodent system, or we expect as a result of work in the rodent system, that our human neurons are going to show a decrease in their overall dendrite complexity as well as a decrease in their soma size. And so we were very interested to determine if this electrophysiological phenotype could be correlated with morphological phenotypes in our iPS cell-derived neurons. In order to facilitate the analysis of dendrite morphology in our cultures, we adapted protocols for being able to label individual neurons with, in this case, GFP. We just used a modified transfection protocol for being able to label at relatively low efficiency neurons with a very, very strong transgene in order to completely fill the neuron, allowing us to then trace the morphological complexity of the cell. And when Wesley Lai, a student in the Ellis lab, performed these experiments examining control neurons and neurons from individuals with Rett syndrome, Wesley found when comparing multiple MECP2 expressing cell types versus cells from individuals with Rett syndrome, that there was a pronounced decrease in the total dendrite length of these neurons, as well as in the complexity of these neurons, as revealed by dendrite tip number and is shown here in a shoal analysis. And in addition to a decrease in total dendrite length, we've also found by work in the Ellis lab, amongst other groups, is that there's a decrease in the size of the actual cell body of neurons, or the soma. So this is an experiment from the first paper from the Ellis lab modeling Rett syndrome, where Aaron Chung, a student in the lab, generated isogenic lines that either did not express MECP2 or expressed MECP2, and these were generated from the same woman with Rett syndrome. And what Aaron found was that there is a pronounced decrease in the overall size of the soma in the isogenic mutant versus control cells. Importantly, this has been recapitulated using multiple wild-type lines as well as multiple lines from a second individual with Rett syndrome. And this difference in soma size is consistent regardless of a number of different experimental parameters that we've examined using slight modification of our usual neuronal differentiation protocol from Bernand and Gage, as well as by including DAPT, which will force neural precursors to exit the proliferation phase and to become neurons more swiftly. Regardless of the protocol that we use, we still see this quite consistent, approximately 25 or 30% decrease in soma size, comparing wild-type cells to cells from individuals with Rett syndrome. And importantly, Aaron Chung and Ugi Juric did experiments in order to examine whether these phenotypes could be rescued. In this case, they used lentivirus vectors that encoded the MECP2 transgene with a MIC tag on it. When these neural precursors were infected with the lentivirus, some of the cells became infected and some of them were not infected. And by comparison of the MECP2 expressing cells to the MECP2 null cells, what they found was a robust rescue of the soma size deficit that was observed. And importantly, this argues that this deficit is a cell autonomous defect, where by expressing MECP2 within the individual cell of interest, we rescue the phenotype, but only in those cells. And so essentially what these data and other data have shown is that iPS cells have been generated from individuals with Rett syndrome, that these iPS cells have been differentiated into cells with a cortical identity, and that neurons from individuals with Rett syndrome exhibit disease-associated phenotypes as expected from work done in the mouse, including decreases in the activity of excitatory synapses, the firing of action potentials, the capacitance of the neurons, the length and complexity of dendrites, and in soma size. And most importantly, this soma size deficit was rescued by reintroduction of an MECP2 transgene. And so some of the future directions that we and many other people are interested in pursuing are, for one, drug screening for Rett syndrome. We've found in a number of different experiments that soma size is consistent and it's relatively easy to measure and that it can be rescued, suggesting that it could, or other related phenotypes could be used as the foundation for developing drug screens. And the Ellis Lab, amongst others, are pursuing this, and we are currently developing tools aimed at developing these screening conditions. And now that we and others have found that neuronal phenotypes have been identified in neurons from individuals with Rett syndrome, we're very interested in expanding this work. And we are currently focusing on performing phenotypic analyses of neurons from individuals with autism spectrum disorder, amongst other disorders. We have generated iPS cells from multiple individuals who have mutations of high-priority autism candidate genes. And in collaboration with Dr. Stephen Scherer's lab at the Hospital for Sick Children, we're also performing genome editing with CRISPRs in order to introduce or correct ASD-associated mutations in our iPS cells. And I'd just like to thank the people who did this work. Aaron and Ugi were PhD students in the Ellis Lab and took the lead on these projects that I described today. Rebecca, Wesley, and Jason also performed important contributions to these studies, and many of the neurons that were generated were done by Alina Piekna and Wei Wei. Dr. Michael Salter is an essential collaborator in this study, and his postdoc Wenbo Zhang performed all the electrophysiological experiments, and Dae-Sung Kim, who now has his own lab in Korea, was involved in some of the differentiation protocols. And we need to thank our funding agencies, the CIHR, the NIH, the Province of Ontario Neurodevelopmental Disorders Network, and the Ontario Stem Cell Initiative, who provided my fellowship support. And finally, the work that was discussed in the previous slides can be found in these citations, and if this is work that you are interested in pursuing or that you're interested in reading more about, I highly recommend a number of other studies, but for the sake of space, I've limited to some of these studies published by a number of groups, including Ricardo Dolmesh's lab, Fred Gage's lab, that performed some really, really exciting disease modeling using this approach.